Hello everyone, I am Yugal Bindari, PhD student at University of New England. I would like to welcome you all on my presentation. The title of my talk today is Molecular Detection of Eimeria species and Clostridium perfringens in poultry dust and pool etc. of commercial broiler chicken flocks differing in productive performance. Introduction. Necrotic enteritis and coccidiosis are economically detrimental enteric diseases that are usually associated. Coccidiosis is usually caused by 5 species of Eimeria, whereas Clostridium perfringens type A carrying NET-B gene is the most virulent strains capable of causing necrotic enteritis. Monitoring of both diseases on commercial flock is usually based on gut gross pathology, which is subjective, requires skilled personnel, and has no predictive value. Therefore, in our study, we evaluated the usefulness of population-level samples such as dust and pooled etc. with an aim to answer 3 research questions. Can population-level samples such as dust and pooled etc. be used to monitor Clostridium perfringens, NET-B, and Eimeria species in commercial broiler farm? Can dust be used as an alternative to etc. samples to detect Clostridium perfringens, NET-B, and Eimeria species genome copies in commercial broiler farms? Can high and low performing farms be differentiated based on the genome copies load of Clostridium perfringens, NET-B, and Eimeria species in dust and pooled etc.? Methods. The study was conducted in 16 flocks of 8 commercial broiler farms in a 2 by 2 by 5 factorial arrangement of treatments with 2 companies, company A and B, 2 farm productive performance, high and low performing farm, and 5 sampling times, 7, 14, 21, 28, and 35 days of chicken age. In this study, veterinarians from each company selected 2 high and 2 low performing farms based on the past production data. So total number of farms were 8 in this study. Each farm had 2 flocks. So total number of flocks were 16 from where the samples were collected. You can see here this is etc. samples. Etc. samples were collected from the floor of the poultry house. 5 individual etc. samples were collected from poultry house to make 1 pool sample, and 4 pool etc. sample were collected from each poultry house. You can see here this is dust collection jar. Dust samples were collected using dust collection jar. Dust collection jar were suspended at 1.5 meter height from the ground from each poultry house. 2 dust collection jar were suspended at 1.5 meter height from the ground in each poultry house. So 2 dust samples were collected from each poultry house. Dust samples and etc. samples were collected at days 7, 14, 21, 28, and 35. DNA extraction of the samples were done by using QAM fast DNA stool minikit with modification. Repeated methods ANOVA was performed to determine the overall load of the pathogen. General linear model was performed to determine the shedding profile of the pathogen across each farms. Intra-class correlation coefficient was performed to determine the level of agreement between dust and etc. genome copy load for each pathogen. All the data analyses were carried out in GMP software. Results. Detection profile of Eimeria osserbulina genome copies. Here in the figure, A1 to A4 refers to company A farms and B1 to B4 refers to company B farms. Solid line in the figure denotes high performing farm and dashed line in the figure denotes low performing farm. In etc., all farms were positive for Eimeria osserbulina. Eimeria osserbulina genome copies load in etc. peaked at days 21 and 28 and decreased at day 35. In dust also, all farms were positive for Eimeria osserbulina. Eimeria osserbulina genome copies load in dust peaked at days 21 and 28 with no further decrease at the last measurement. Intra-class correlations showed excellent agreement between dust and etc. genome copy load in case of Eimeria osserbulina. Detection profile of Eimeria maxima genome copies. In etc., all farms were positive for Eimeria maxima. In company A farms, Eimeria maxima genome copies load in etc. peaked at day 21 or 28 and then decreased for farms A1 and A4 at day 35. In company B farms, Eimeria maxima genome copies load in etc. peaked at day 35. In dust also, all farms were positive for Eimeria maxima. In company A farms, Eimeria maxima genome copies load in dust peaked at day 28, whereas in company B farms, Eimeria maxima genome copies load in dust peaked at day 28 or 35. Intra-class correlations showed excellent agreement between dust and etc. genome copy load in case of Eimeria maxima. Detection profile of Clostridium perfringens genome copies. In etc. Clostridium perfringens genome copies were detected at day 7 in most of the farms and then declined afterwards, except farm B2 in which Clostridium perfringens genome copy load in etc. peaked at day 21 and remained elevated afterwards. In dust, all farms were positive for Clostridium perfringens. In company A farms, Clostridium perfringens genome copy load peaked at day 14, whereas in company B farms, Clostridium perfringens genome copies load in dust peaked between days 7 and 21. You can see here detection profile of Clostridium perfringens genome copies load in etc. and detection profile of Clostridium perfringens genome copies load in dust were completely different. An intra-class correlation also showed no agreement between dust and etc. genome copies load in case of Clostridium perfringens. Detection profile of NetB. In our study, NetB was only detected in one high-performing farm. In both dust and etc., detection of NetB peaked at day 21 and remained elevated. Intra-class correlations showed excellent agreement between dust and etc. genome copies load in case of NetB. In our study, all farms were positive for Clostridium perfringens, Eimeria osserbulina, and Eimeria maxima, whereas only one low-performing farms from company A was positive for Eimeria brunetti in etc., and all farms were negative for Eimeria tenella and Eimeria necatrix. Neither of our farms were detected positive for Eimeria tenella and Eimeria necatrix. Overall, the study found no difference in pathogen genome copies load between high and low performing farms. You can see here, this is the figure for the dust and this is the figure for etc. This is high performing farm and this is low performing farm. In dust, overall, Eimeria osserbulina, Eimeria maxima, and Clostridium perfringens genome copy load were similar in high and low performing farms. Similar results were seen in etc. Also in etc., the study found no difference in genome copies load of Eimeria osserbulina, Eimeria maxima, and Clostridium perfringens between high and low performing farms. Usually in case of Eimeria, an oocyst count of more than 50,000 per gram of pooled etc. has been associated to loss of performance. However, in our study, Eimeria genome copies load per gram of pooled etc. was lower than 50,000, which indicates that none of the flocks in our study were at risk for the loss of performance. From the results, we conclude that dust and pooled etc. could be used to detect Clostridium perfringens, NetB, and Eimeria species in commercial boiler farms. Dust can be used as a proxy for etc. samples to determine Eimeria osserbulina, Eimeria maxima, and NetB, but not for Clostridium perfringens. High and low performing farms had overall similar genome copies level of Eimeria osserbulina, Eimeria maxima, and Clostridium perfringens, and this might be due to low to moderate genome copy load of the pathogens detected in our study. Acknowledgements. We are thankful to AgriFutures Australia for funding this project. We are also thankful to our lab technician Daniel Smith for providing technical assistance during the laboratory analysis. We are also thankful to farmers and veterinarians from the company who participated in this study. Thank you.